Hi, this is Matt Dean with A Plus College Ready, and we're going to talk a little bit today about evolution. First of all, a little bit about the history of the theory of evolution. We'll start with Carolus Linnaeus. Carolus Linnaeus was the father of taxonomy. That's the science of classification. In his original system, he organized all of life into two kingdoms, the plant kingdom, or plantae, and animalia. He used a system called binomial nomenclature. This means that each organism was assigned a two-part name. In binomial nomenclature, the genus name is written first and has a capitalized first letter. The species name is written second and is not capitalized. The whole name is written in Latin, and it's either italicized if it's typed or underlined if it's written out. The current levels or taxa in the classification system include the largest, most inclusive domain, the domain. Domains are composed of similar, evolutionarily related kingdoms. Kingdoms, on the other hand, are composed from similar, evolutionarily related phyla, or in plants, we call those divisions. So we can see there are three domains. There are bacteria, there are archaea, both composed of prokaryotic cells, and then there are eukarya. Eukarya, that includes animals, plants, fungi, and protists. So here are the kingdoms. Depending on the classification system that you look at, you may hear about 5, you may hear 6 kingdoms. The one that's sort of debatable is Protista, because this one includes both the animal-like protists like euglenas and amoebas. It also includes the plant-like protists like algae. So a phylum or a division, these are composed of similar evolutionary related classes. And then next comes class, then order, family, genus, and finally species. Species is the least inclusive, most specific taxon. So species, again, is the least inclusive of all categories. It's the most specific taxon. Organisms in the same species are very closely related. They can interbreed, and their offspring are fertile. An easy way to remember the order of the taxa is using an acronym. There are lots of different ones. One of the most popular ones right now is this one. Dominating King Philip came over for green salad. So again, that's domain, kingdom, phylum, class, order, family, genus, and species. So Linnaeus originally based his taxonomic system on morphology, like body shape and structure. But the modern classification system is based on evolutionary relationships. Organisms in the same taxa are put there because they're thought to be evolutionarily related and share common ancestors. So here we can see sort of how the system works. Now, domain would be up here. So we're looking at humans. So humans would be in the domain eukarya. That would be the same domain as plants, protists, fungi, and all other animals. But we're in the kingdom Animalia. So that eliminates plants, fungi, protists. We're in the phylum Chordata. This is essentially the vertebrate animals. So all the invertebrates are eliminated at this point. We're in the class Mammalia. So this includes only animals with that are vertebrates, that feed their young with milk, have body hair, are warm-blooded. We're in the order Primates. So these are essentially mammals that have 3D vision and an opposable thumb. Essentially, we're talking about the monkeys here and the humans and the monkey relatives like the apes. Then we get to the family Hominidae. These are primates that can walk upright. We get to the genus Homo. So these include modern men and some of what we might call cavemen, like Homo habilis and so on. And then finally we get down to Homo sapiens, which is our specific species for modern human beings. And we are the only organisms in that species. As you can see, we start at the most inclusive category, which is actually domain. And then as we go down, the number of organisms in each taxa get less and less because the organisms in those groups are becoming more and more related. Next, we'll talk a minute about Charles Lyell. Lyell became one of Darwin's best friends. He wrote a book called The Principles of Geology. He came up with this idea of uniformitarianism. And essentially, he said that the same geologic processes that were happening in the present have been happening over all of time, and that these processes are constantly changing the Earth. Things like the Grand Canyon was formed by erosion over many, 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 many years. So his ideas were sort of incorporated into Darwin's, and they supported his idea, and essentially they led a lot of support to the fact that the Earth had to be a lot older than people at the time thought it was. Then we get to Jean-Baptiste Lamarck. So Lamarck proposed a theory of evolution through the inheritance of acquired traits. He proposed this theory in 1809. Darwin was just born. A lot of times the giraffe is used as an example for his theory. The main ideas with this theory are these. Number one, living organisms or their component parts tend to increase in size. Production of a new organ occurs when there's a need for a new organ. Continued use of an organ makes it get more developed, while disuse of an organ results in its degeneration. And acquired characteristics or modifications developed by individuals during their own lifetime are inheritable, and they accumulate over a period of time, and these can result in a new species. The problems with the theory are essentially that Lamarck didn't know anything about genetics. Lamarck was before Mendel. He didn't understand genetics. Acquired traits cannot be inherited. They can't be passed on. Just because you work out a lot and get big muscles during your life, that doesn't mean your children are going to be born with big muscles. Genetic traits are passed to the next generation. Acquired traits are not. It's also true that living organisms don't necessarily tend to increase in size. New organs aren't just produced when there's a need for one, and organs that aren't used don't necessarily just degenerate. So there's a lot of problems with Lamarck's theory. Now we'll get to the more accepted current theory, evolution via natural selection. This was proposed by Charles Darwin and also Alfred Wallace in about 1859. Like I said before, it's the current accepted theory of evolution. It's compatible with an understanding of genetics. According to Darwin, natural selection is the mechanism of evolution. Natural selection is the process in which organisms that are best adapted to their environment tend to survive and transmit their genetic characteristics in increasing numbers to succeeding generations, while those that are less adapted either are eliminated or don't reproduce as much, and therefore their genes aren't passed on as often as those that are better adapted to an environment. Evolution, in its most simple form, is about reproduction. Organisms that are better adapted to an environment out-reproduce those that are poorly adapted to that environment. Darwin essentially came up with 4 factors that need to be in place for evolution through natural selection to occur. First of all, there has to be genetic variability in the population. That variation can come through sexual reproduction, things like random fertilization of sperm and egg, crossing over during meiosis, independent assortment during meiosis. It can come from mutations. It can come from immigration. But there has to be variation within the population. Second one, more offspring are produced than can survive. Nature selects certain organisms to die. Certain organisms die because of limited resources, predation, etc. 3, some organisms are better adapted than others, and these adaptations have to be genetic. The organisms that are better adapted are more likely to survive, but according to number 4, the most important thing is that they're more likely to reproduce. And because they have more offspring than their competitors that aren't as well adapted, they become more and more common, and the genes of their competitors become less and less common in the population. Evolution is often referred to as the survival of the fittest. Fitness does not necessarily mean the strongest or the biggest or the baddest. In a biological context, fitness means the ability to survive to reproductive age, get a mate, and produce offspring. It's about reproduction. Organisms that are the most fit have the most babies and therefore pass on the most genes. You might also hear the word fecundity used to refer to the reproductive rate. So here's sort of a comparison between Lamarck's idea and Darwin's idea. So Lamarck's idea was that the ancestors of giraffes had little short necks, and that as time went on, competition happened. There wasn't as much food for them to be able to reach, so they started stretching their necks. And somehow, as they stretched, their neck actually became longer. And over generations, it became longer and longer and longer. And their babies were born with longer and longer necks. Now, again, that sounds ridiculous because we know none of that's genetic. This is about acquired traits. And we know those can't be passed on. Darwin's idea was that there was some variation in the population. There were some short-necked giraffe ancestors, but there were some with longer necks and some with even longer necks. And that because of the limited resources, the lack of food, those with longer necks could reach higher, get more leaves, they had full bellies, therefore they were more healthy, they had more babies, and that over time, the genes for longer necks became more and more common than the genes for shorter necks. And that's why the giraffes surviving today have longer necks. That's natural selection. That's Darwin's idea of evolution. So there's some important terms associated with evolution that you need to be familiar with. First of all, you've got to know this definition of evolution. Evolution in its most simple form is a change in the allele frequency of a population over time. Now remember, an allele is a form of a gene. So we're essentially just looking at how common are certain versions of a gene compared to others in a population over time. It's important to remember that the population, the group of the same species in the same place at the same time, the population evolves. Individuals cannot evolve. That's a huge point to know. There's also what we call microevolution. So microevolution is when evolution happens within a population, usually happens over a short time, and it causes small changes within the population, but not enough to lead to speciation, or the formation of a new species. Macroevolution, on the other hand, is a major evolutionary change within the population that splits it into different species. So sometimes we'll use the word species essentially synonymously with macroevolution. We already talked about natural selection, Darwin's mechanism of evolution. Here's a more formal definition: the process whereby organisms better adapted to their environment tend to survive and produce more offspring. And again, Darwin called that, or people call it today, Darwin's mechanism of evolution. It's how it happens. Another important term to know about is artificial selection, or what's also sometimes called selective breeding. So this is when humans actively choose which traits are passed on to offspring. This could be in crop plants, or it could be in certain kinds of animals that we breed. Even before we knew about genetics, farmers had been choosing which cattle got to breed, which plants bred with which other plants. So they were picking beneficial traits. They knew they could be inherited. And over time, essentially, they caused human-caused evolution. That's artificial selection. So there have been lots of situations in which artificial selection has backfired or caused some negative outcomes. One of those is with insecticides. So as we've used more and more insecticides, insects with genes for resistance or tolerance to the insecticide, those have been the ones that have lived and the ones that have reproduced. So the insects become more and more resistant, more and more tolerant of the insecticide across generations. The same thing has happened with antibiotics. When you use antibiotics, you kill off first the bacteria that have no resistance or very little resistance. And the ones that might get left behind and passed on and able to reproduce at least, those are the ones that are somewhat resistant. And as we use more and more antibiotics for longer and longer periods of time, resistant bacteria have been selected for, and non-resistant bacteria have essentially been killed off. So our antibiotics become less and less effective across time because of this. That's also true for antiviral drugs like those used to treat HIV. The HIV virus has become more and more resistant to those drugs. So let's take a minute and talk about some patterns of selection. And we'll first talk about what's called stabilizing selection. This occurs when individuals at the extremes of a range of a characteristic are consistently selected against. Let's talk about the birth weight of babies before modern medicine. If a baby was really, really small, it was probably kind of sickly and might have died of some kind of infection or just lack of development. If a baby was really, really big, that baby might not have been able to be born. It might have been too big to actually get out of its mother. So those babies were selected against. The babies that were selected for by nature were those that were middle-sized. So over time, most babies became or were born middle-sized, like 6 to 7 pounds. Now, things like neonatal care units, things like the C-section, those are changing those patterns. But as far as stabilizing selection goes, it's when nature chooses an intermediate, a middle phenotype as the one best for the environment. There's also directional selection. This one is when individuals at one extreme of the range of a characteristic are selected for, and those in the middle and those on the other extreme are selected against. So this happens a lot when there's a consistent change in an environment. For example, if you were using a certain insecticide on a field, the insects that had some resistance to that insecticide, those would be selected for. And the insects without that resistance, those would be selected against. So that over time, there would be a huge shift in the allele frequencies in the next few generations, and the frequency of the resistance gene would rise quickly and dramatically in that population. There's also what's called disruptive or diversifying selection. This happens when both extremes for a characteristic are selected against, and the intermediate is selected for. This can happen in lots of different places. For example, there are many kinds of insects that feed on leaves of trees. A lot of these have colors that match the leaves they feed on. Suppose that a species of insect ranges in color from light green to dark green, but medium green is the most common. If a particular species of insect had some individuals that fed on plants with dark green leaves, whereas other individuals fed on plants with light green leaves, medium green insects would be selected against, and the two extremes selected for. So here we see some examples. So this top example is showing us stabilizing selection. So we started off with this kind of distribution, but for some reason in this environment, the middle phenotype was selected for. So over time, that phenotype became more and more common, and these phenotypes out here on the extremes became less and less common. With directional selection, the middle one here. Here we're looking at the example of the peppered moth. In the Industrial Revolution, the smoke, the soot from the factories got all over the trees, made the trees darker than they normally would. So dark moths, which had been rare before the Industrial Revolution, they were selected for. The normal phenotype, which is kind of mottled, spotted almost, those became selected against because things like birds could see them. So very quickly, the frequency shifted from this blue pattern that we see here in the original population to the red pattern. So black was selected for. So the diversifying or disruptive selection, the last one here, we're looking at some rabbits. the Himalayan rabbit, the gray and white, those are able to blend with rocky environments. Also, well, let's look at the original population here. So the original population was mostly white, with some that were almost a dark gray, and then some that were part white and part gray. In the new environment, these guys could blend in with rocks. So could these. The white ones could not. So those guys were eaten. So the frequency of the population very quickly shifted so that the dark gray became more and more common. The gray and white became more and more common. But the white rabbits themselves, those became less and less frequent. So that's diversifying or disruptive selection. So now let's take another sidebar and talk a bit about population genetics. Population genetics essentially says that populations evolve, individuals don't. It's the science that studies the trait variation rates over time within a population. It's basically looking at allele frequencies in the gene pool of a population. Remember that a population is defined by 4 main criteria. First of all, they have to be members of the same species of organism. They have to be located in the same place at the same time, and they have to show signs of reproduction. Those are the characteristics of a population, a biological population. So now let's talk about Hardy-Weinberg equilibrium. It's also called genetic equilibrium. It's a theoretical condition in which a population's genotype and allele frequencies remain unchanged over successive generations. Essentially, it's when evolution is not occurring within the population. Now, this is theoretical, probably never completely happens, but according to in order for Hardy-Weinberg equilibrium to be achieved, there are 5 requirements that must apply to the population. First one, there can be no mutations. Because remember, a mutation is a new allele. So just when a mutation happens, allele frequencies have changed because it's a new allele. Especially important are germ cell mutations. These are mutations that happen in the gametes or the cells that become the gametes. These are mutations that can be passed on to the offspring. Mutations that happen just in somatic cells or normal body cells, those aren't passed on. Those aren't important, at least not to evolution. The second requirement is there can be no immigration, either incoming or outgoing. Because immigrants who either coming in may bring new alleles, or those going out may take out with them unique alleles and change the frequencies within the population. Sometimes instead of saying no immigration, sometimes we could say there can be no gene flow between different populations. Number 3, the population has to be really large in order to avoid what's called genetic drift. Genetic drift is a type of unpredicted change in allele frequency that happens just due to chance. It usually happens only in small and very isolated populations. The next one, there can be no natural selection. Essentially, this means that all organisms have an equal chance of surviving. And then lastly, there can be no sexual selection. This means that all organisms have an equal chance of mating. Essentially, another way of saying this is that mating must be random. So the Hardy and Weinberg developed some equations that can be used to calculate the genetic variation of a population when it's at equilibrium. It's important to note that their equations can only be used within populations that are at genetic equilibrium. So they came up, this is the basic Hardy-Weinberg equation. And we also are going to deal with this one right here. In this equation, P stands for the frequency of the dominant allele. Q stands for the frequency of the recessive allele. Together, those frequencies, which you want to work out in decimal form, add up to be 1. And then in the major Hardy-Weinberg equation, this one, P squared is the frequency of the homozygous dominant genotype. 2PQ is the frequency of the heterozygous genotype, and Q squared is the frequency of the homozygous and recessive genotype. Those three all have to add up to be 1. So the Hardy-Weinberg equations are used to calculate the allele frequencies of a population. And if you compare those allele frequencies across generations, you can see if evolution is happening. We have a whole separate video on Hardy-Weinberg equilibrium and how to work problems associated with it. So make sure you see that video. Let's talk a brief minute about genetic drift. Remember, genetic drift is a random fluctuation in the frequency or appearance of a gene in a small, isolated population. It's usually caused by chance, not natural selection. So we're going to talk briefly about 2 types of genetic drift. The first one is called the founder effect. This happens when a new colony is started by just a few members of an original population. This small group, the small founding group, may have reduced genetic variation compared to the original population, and they probably have a non-random sample of the genes in the original population. They may have more of a certain kind of allele than the main population. So a good example of this is the Afrikaner population of Dutch people that settled South Africa. They started as only a very small group of colonists. But today that population has an unusually high frequency of the gene that causes Huntington's disease. And that's because the original founders, several of them probably carried this gene or had this gene, and they had it at a much higher frequency than the normal Dutch population. There's also the bottleneck effect. This is when genetic drift results from the reduction of a population due to some kind of natural disaster or maybe some kind of human activity. So that the new population, the organisms that are left, they're not necessarily representative of the original population. For example, the northern elephant seals have very little genetic variation today, probably because of a bottleneck that humans inflicted on them in the 1890s. We hunted them almost to extinction, down to about 20 individuals. Today, there's over 30,000 of them, but their genetic variation is very low because all 30,000 of those descended from the 20 that were left behind after human hunting. So another topic that we'll address briefly here is speciation. First of all, let's think about the biological species concept. A species consists of genetically similar organisms that interbreed and produce offspring that are fertile. They can have babies themselves. For example, horses and donkeys are very closely related, but they're not in the same species, even though they can reproduce and have a baby, a mule. Horses and donkeys are in separate species because that baby, the mule, is sterile. So even though they're closely related, they're not in the same species. So for speciation to occur, for speciation, the formation of new species to occur, first of all, you start out with 2 populations of the same species, and in some way or another, those 2 populations are isolated from each other. They're reproductively isolated. That means there's no flow of genes between them. Over time, random mutations happen in both groups. Since they're random, it's probably different mutations that happen in those 2 groups. Some of those mutations are good and selected for. Some are bad and selected against. But over time, those mutations cause allele frequency changes to happen. And given enough time, this process can cause those separated populations to diverge into different species. So we're going to talk about 2 or 3 main kinds of speciation. The first one is allopatric. This is when you have 2 populations that are separated by some kind of geographical barrier, maybe an ocean or a mountain or a huge ravine. Because they're geographically separated, they can no longer reproduce. Random mutations happen. Different mutations in the 2 populations. Some are selected for, some are against. And over time, more and more mutations, more and more differences accumulate. Eventually, so many mutations and so many events happen that the two species are no longer, the two populations at least, are no longer the same species. They're two different species. Now there's also sympatric speciation. This is when two populations live in the same geographic area, but are still reproductively isolated. This happens more commonly in plants. And it often happens when a plant, something goes wrong in meiosis or in reproduction, and a plant becomes polyploid. It gets an extra set of chromosomes. So it can no longer reproduce with maybe the diploid plants that it descended from. And because they can't reproduce together anymore, those two populations, again, just like in allopatric speciation, build up random accumulations of mutations, and over time, the two populations become more and more different, so different that they're not the same species anymore. The last type of speciation we'll mention is called parapatric. So this occurs when populations are separated not by a geographical barrier, but by some kind of extreme change in habitat. So they might be living side by side, and they ought to be able to interbreed, but usually they don't. Good example of this are plants that live around mines with really contaminated, toxic soil. The plants that live in that soil, a lot of them died, but the plants that survived were the ones that were more tolerant to the metals, the heavy toxic metals in the soil. Well, living just adjacent to that polluted area are probably plants of the same species, but since that ground isn't polluted, the same kind of selection didn't happen there. So you would think that those two plants would interbreed, but oftentimes they don't. And something that causes them not to interbreed are mutations. In some cases, a mutation might happen that causes the flowering time of the plants in the polluted soil to change, a type of temporal separation, so that they no longer can interbreed. Their genes can no longer flow with the plants that are living just maybe a few feet away in the non-polluted soil. Again, this causes random mutations to accumulate, differences accumulate across time, until eventually those two groups, those two populations, are no longer the same species. We'll also talk briefly about adaptive radiation. So this is a process in which organisms diversify very rapidly from some ancestral species into a multitude of new forms and probably new species. And this really happens a lot when there's a change in the environment. Oftentimes it happens after some kind of extinction event, maybe, where a lot of new open niches are available. So it starts with a recent single ancestor with lots of speciation, and it can occur due to a combination of allopatric, parapatric, and sympatric speciation events. Darwin's finches are often used as examples of adaptive radiation. So the founder species might have looked something like this. But due to limited resources, due to different kinds of speciation events, that founding species very quickly speciated into all these different species with different kinds of beaks that could access many different kinds of food. And here we can see maybe it even closer, how that the original ancestral seed-eating ground finch very quickly speciated out into all these different forms of finch with all these different forms of beaks that could access many, many different kinds of foods, and therefore avoid competition with the other kinds of finches. Again, that's called adaptive radiation. We'll also talk very briefly about what are called reproductive isolating mechanisms. These are a collection of evolutionary mechanisms like behaviors or physiological processes which are critical for speciation. They prevent members of different species from producing offspring, or they ensure that any hybrids that are formed are sterile. These processes help to maintain the integrity of a species by reducing the flow of genes between species that are related. They're usually categorized as either prezygotic, before the formation of the zygote, or postzygotic, after the formation of the zygote. The prezygotic oscillating mechanisms prevent related species from ever forming zygotes. Here's some examples. Habitat oscillation. Good example of this are lions and tigers. They are closely related. They don't typically form zygotes, though, because they're living in very different parts of the world. They don't come into contact with each other in nature. Another example is behavioral oscillation. Most animals have some type of mating dance or mating ritual, mating behavior. And organisms from different species may have different mating dances or different mating behaviors. So they're not attracted to each other. Their mating behaviors don't allow them to ever come together to try the physical act of mating. There also can be temporal or time oscillation. Related species may have different breeding seasons, or like in flowers or plants, they may have different flowering times. So they can't reproduce because their mating seasons are different. There's also mechanical oscillation. This just essentially means that the reproductive parts of these two species no longer fit together and work together correctly. And lastly, there's gametic oscillation. This means that even though the two species may be attracted to each other, they may go through the physical act of sex, but when the sperm and eggs get together, that they can't form a zygote. They don't recognize each other and can't form a zygote. So these prezygotic oscillating mechanisms prevent related species from forming viable zygotes. And then there's also postzygotic oscillating mechanisms. These are mechanisms which act after fertilization, but they prevent successful interpopulation or interspecies production of viable offspring. Maybe the most famous one of these is reduced hybrid viability. So even though these two species can mate, they can form a zygote, they can form an offspring, it just lacks the ability to live. It has reduced survival ability. There's also reduced hybrid fertility. This would be the mule would be a good example of this. Horses and donkeys, different species, but they can mate. But their babies, the mule, is sterile. So a mule itself is not really a species because it can't reproduce itself. All right, another topic that we'll mention here briefly is evidence for evolution. And a key word is the word phylogeny or phylogenetics. This is essentially the evolutionary history of a species. What are its ancestors? What did it evolve from? So let's talk about some pieces of evidence. Let's talk about homologous features, or we might also call them homologies. These are structures in different species that are similar because they share common ancestors. Think about the arm of a human, the wing of a bat, the flipper of a whale. The DNA that codes for all three of those structures is very, 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 very similar. That's why those structures are similar. And the DNA is similar because it all came from a common ancestor shared between the human, the bat, and the whale. So DNA is sort of the ultimate homology. It's the shared genetic code of essentially all of life. Shared RNA, shared protein structure, all of those can be thought of as homologies. But if asked to give the most compelling evidence of evolution, the similarity of DNA sequences across life is by far the best evidence. Now, there are also these structures called analogous features. These are similarities in two species due to convergent evolution, rather than due to being descent from a common ancestor. Think about the wing of a bird and the wing of a mosquito. Yeah, birds and mosquitoes can both fly, but their wings are very, very different. They're coded for by very, very different DNA sequences. So even though birds and mosquitoes both fly and they both have wings, this does not imply they have common ancestry, because the genes that gave both of them the ability to fly are very different sets of genes. This indicates nature finding different solutions to the same evolutionary problem. Now, there are also structures called vestigial organs or vestigial structures. These are structures that are sort of a historical or evolutionary remnant of a structure that was important in evolutionary ancestors. Think about the appendix in a human or the pelvis in a whale. We think whales have a pelvis because their ancestors were four-legged, land-walking mammals, and that they still have that pelvis and maybe some small leg bones internally. Snakes, the same way. They have a vestigial pelvis and vestigial leg bones because we think their ancestors were four-legged, lizard-like animals. The fossil record is another really important source of evidence for evolution. No matter where you go on Earth, in the deepest layers of the rock, the oldest layers of the rock, where you can find vertebrate fossils, you only find fish. And as you come up, you'll find layers where you find only fish and amphibians. And then as you come up some more, you'll find levels where you'll only find fish and amphibians and reptiles. And then as you come on up some more, you'll find levels where you find fish, amphibians, reptiles, and then birds and mammals at about the same time. That pattern is the same all over the world, no matter where you go. And scientists would say that that's evidence that the first vertebrates were fish, and that some fish-like, some amphibians evolved from fish-like ancestors, and reptiles evolved from amphibian ancestors, and birds and mammals evolved from reptile ancestors. In addition to the fossil record, we also sometimes use what's called comparative embryology. So this is studying the similarities and differences amongst how embryos develop. And the argument is that the more similar patterns of embryonic development, the more related organisms are. We already talked a little bit about this one without naming it: comparative biochemistry or comparative molecular biology. So this is comparing either the DNA or the RNA or maybe the protein amino acid sequences, and also even metabolic pathways of related organisms. And organisms which share more of those things are more related to each other than they are to organisms in which there are more that have more differences in those things. Another kind of evidence for evolution that Darwin even cited was artificial selection. And his argument was essentially, if man can make evolution happen, so can nature. The last couple of pieces of evidence for evolution we'll talk about is just, first of all, direct observation of microevolution. We've seen resistance to antibiotics and resistance to pesticides happen within our lifetimes, within the last 75 years. Documented cases of real evolution happening. We'll also mention biogeography for a second. So looking at where organisms live on Earth and why they live there. Let's talk a little bit about marsupials and other kinds of mammals. We think probably the first mammals on Earth were monotremes, like the egg-laying mammals. And then at some point in time, from those monotreme ancestors, marsupials, pouched mammals, developed. Sometimes in that area, Pangea split into different pieces. And that the marsupials, at that point in time, would have been the only kinds of mammals. Sometimes after that split, placental mammals, non-pouch mammals, evolved. But they didn't evolve, those mutations that caused them to happen didn't happen in Australia, which was separated by oceans from everything else. So the placental mammals outcompeted the marsupials in most of the rest of the world, and most marsupials were extincted, with a few exceptions, like the possum. But that never happened in Australia because the mutations that led to the development of placental mammals never happened there. So the marsupials continued to live there, continued to thrive. So those kinds of cases of organisms being found in only certain places, and the geographic explanations for why are also good pieces of evidence for evolution. Here we see a comparison of the amino acid sequences in hemoglobin, one of the proteins in your blood, main one that carries oxygen for us. So if you compare human amino acid sequences in the hemoglobin protein to other humans, there's zero differences. Compare that to monkeys, there's 8 amino acids different out of 146 total. With mice, there are 27 different out of 146. Chickens, 45 out of 146. Frogs, 67 out of 146. And lampreys, a type of jawless fish, 125 out of 146. So scientists would say this means that humans are most related to monkeys, and then still pretty closely related to mice, more distantly related to birds, even more distantly related to amphibians, and even more distantly related to fish. And notice that this evidence follows the same pattern that we talked about with the fossil record. Fish first, then amphibians. Reptiles aren't on here, but then reptiles, then birds, then mammals, and then somewhere later in mammal evolution, humans. We'll also mention what are called the patterns of evolution. Darwin had this idea of gradualism. His idea was that evolution happens very slowly. So there's small variations in which certain organisms are a little bit better adapted to an environment than are others. Organisms with those genes are selected for. They have more babies and pass on more genes than individuals that don't have those genes. And this causes a very gradual, slow change in the population across time. It's slow, constant, consistent. There's also, though, an idea of punctuated equilibrium. This is when change happens very fast, in a very big way, in spurts. So there's a period of very little. There are long periods of very little change. That's the equilibrium part of the term punctuated equilibrium. And then some kind of natural disaster or plague happens, some kind of cataclysmic event. And then very fast changes happen. There's lots of speciation. There's lots of evolution. And then once the environment stabilizes again, there's very little change until the next cataclysmic event. So today, scientists think that evolution probably happens with a combination of these two ideas. That yes, gradualism is always going on, but that punctuated equilibrium happens when there are these big cataclysmic world-changing events. So the last topic we'll talk about, we mentioned the word phylogeny a minute ago, and we said that it's the history of the evolution of a species or a group, especially in reference to the lines of descent and in relationships among organisms. The ways to establish phylogenetic relationships between organisms, first of all, include comparing the DNA or RNA sequences between the groups, comparing the amino acid sequences of specific proteins, or looking at the morphology, the body structure, shared derived traits. So oftentimes phylogenetic relationships are depicted in what are called phylogenetic trees or cladograms. So a phylogenetic tree is a branching tree-like diagram. Each node or point of divergence has 2 branching lines that come out, indicating an evolutionary divergence from a common ancestor. So a node is a point on that tree where a split happens. So that's a node. So we're saying that there's a common ancestor here, and that some of that common ancestor's descendants became, maybe we'll call this species A. Some of its ancestors became species B. In a phylogenetic tree, the length of the branches between each other represents time. Longer branches mean more time. On the other hand, a cladogram is very similar. But in a cladogram, length doesn't matter. Length doesn't indicate time. Other than that, phylogenetic trees are essentially the same. In AP Bio, we're going to use those terms to essentially mean the same thing. Phylogenetic tree and a cladogram. Think of it like an evolutionary family tree. So there's several important terms that you need to know related to cladograms. First of all, a clade is a group of a biological taxa, maybe like a species, that includes all the descendants from a common ancestor. The root is the initial ancestor common to all the organisms in the cladogram. This is going to be the point which begins the cladogram. Morphology, we said a minute ago, is a branch of biology that deals with like the formation, the body structure of an organism. The ancestral trait is a trait shared by a group of organisms as a result of descent from some kind of common ancestor. A derived trait is a trait present in an organism but absent in the last common ancestor of the group. An outgroup is a group of organisms that serves as sort of a reference when we're drawing a cladogram. Essentially, the outgroup is hypothesized to be less closely related to the in-group than the in-group is to itself. And we'll see an outgroup in our phylogenetic tree in a minute. A node is a point on a cladogram where a split occurs and different species branch off from a common ancestor. It's important to note that in cladograms, the branches of the cladogram can be rotated around the node without changing the evolutionary relationships depicted in that cladogram. And then finally, a clade or a monophyletic group is a common ancestor and all of its descendants, all of the branches and nodes that branch away from that common ancestor. So here we can see a phylogenetic tree. So back here would be the root. This is the outgroup. This is the one that's most different from all of the other species. So this would be a species, species A, species B, species C, species D. This cladogram is showing us that they all evolved from some common ancestor back here. Here's a node. At this node, there's a common ancestor. Some of its ancestors evolved into species B. Some evolved into C and D. Here's a common ancestor shared between C and D. Some of its ancestors became species C. Some evolved into species D. Again, we can sort of see it drawn a different way down here, but the same relationships. So we've got a root. Here's a node. There's a common ancestor there. Some of that ancestor's progeny became species A. Some became species B. Right here's a node. Some of this common ancestor's progeny became C. Some went on to become either A or B. And again, here we can see the root, the beginning of the cladogram, the original common ancestor of everything else. Nodes are points where speciation happens. And again, some of the branches, we can hear them referred to as branches. We can see them drawn in this pattern or this more tree-like pattern. But wherever we go, there are nodes. And remember, nodes indicate the presence of a common ancestor. Now, the first cladograms were drawn based on morphology, based on the presence of shared physical characteristics. And you're going to do an activity later where you use morphology and a table called a character table to construct a cladogram based on morphology. But that's more the old way to draw a cladogram. Today, remember that our taxonomic system is based on evolutionary relationships and DNA. So today, most cladograms are built based on DNA comparisons. So they find certain genes or certain areas of introns and they compare the sequences. The reason that's a good idea is that essentially all organisms use DNA and RNA as their genetic material. It's universal. This means that the nitrogenous base sequences, that the sequences of A's and T's and G's and C's can be compared. Over the courses of millions of years, mutations accumulate. So that means the further back two species separated from each other, there's been more time for mutations in those genes to accumulate. So the more differences, that would mean the less related two species are. The less differences, that would mean the more related they are. Oftentimes when scientists look at molecular sequences, they use the non-coding gene sequences or the intron sequences. And the reason they choose those is because mutations in the genes oftentimes lead to changes that can cause death. And so the mutations aren't passed on as often in the coding, the exons. But the changes in the intron sequences aren't usually as severe. Organisms can live. Mutation rates are higher there. And we said a second ago, in addition to looking at gene sequences, like nucleotide sequences, you can also look at amino acid sequences. These change more slowly, though, than the gene sequences do, because remember that there are several codons that code for each amino acid. The genetic code is degenerate. So that means that there can be mutations without actual changes in the protein. So the best way to create a cladogram is to compare DNA sequences. More differences between the nucleotide sequences mean less related. Less differences mean more related. And again, you're going to do an activity in class where you build a cladogram based on both morphology and on molecular evidence. All right, that's it for our evolution discussion. Hope